Hello everyone, my name is Sanjeev. I work for Novartis in the franchise Neuroscience dealing with the products of multiple sclerosis, and I'm currently employed by a third-party service provider to work for Novartis, but I'd be very happy to work directly at Novartis. And today in this presentation I've taken a topic which is related to my work, but of course it deals about the vaccine development strategies which are existent in multiple sclerosis. With this attempt I wanted to demonstrate how I can present a scientific topic, and that too using digital media because of the current situation of COVID-19. Face-to-face encounters are quite limited, and I would like to take this opportunity to show my motivation and also give a few hints on how I have been working in few different roles in my career and what my education background is. So we'll go into the topic now. So in this talk, the first part is MS therapies approved and under research. Here I'll detail very shortly about all of the therapies which are existent for MS, and in the second step I will talk about which areas of the pathophysiology in MS can we further look upon to develop new treatment strategies, and here how tolerance inducing therapeutics can revolutionize the way we treat multiple sclerosis. And as a special note, I will detail a little bit about how cation homeostasis is being regulated in multiple sclerosis, because it is also a topic of my PhD. And finally, what I bring in addition along with these skills, and why am I specifically interested to work directly at Novartis. I've also detailed or have listed the terms and abbreviations which are used in this presentation. I thought it could be useful for your further reference. So if we look at the overview of the approved therapies, interferon beta has been in the market as a treatment for MS for quite a long time, alongside glatiramer acetate also. They work differently by their mechanism of actions, but both of them have resulted in reducing the annual relapse rates to a significantly nice level. However, since they are old therapies, their efficiency comparing to the new ones is quite less, and also they bring some adverse effects like liver toxicity, lymphadenopathy, and chemotherapy option like mitoxantrone, which inhibits the proliferation of B T cell and macrophages by intercalating with their DNA repair mechanism, is also quite good in reducing the annual relapse rates, but owing to the fact that it's chemotherapy, it induces leukemia and liver toxicity, also cardiotoxicity. The new age disease modifying therapies, which I'll be calling from further on as DMTs, for example fingolimod and siponimod, which are our products, they are sphingosine 1-phosphate receptor modulators. They prevent the egress of immune cells from the lymph nodes, thereby dampening the immune system and modulating the system, thereby causing protection in multiple sclerosis. They are quite good in reducing the relapse rates, annual relapse rates. And teriflunomide, on the other hand, is an enzymatic inhibitor, and dimethyl fumarate works as a redox regulator, and cladribine, being a purine analog, causes B T cell depletion completely. All of these DMTs are very good in reducing the annual relapse rates, but their adverse effect profile is also very notable one, like bradycardia, infections, liver toxicity, lymphopenia, and PML risks, for example for dimethyl fumarate, are very high, and cladribine, being a genetic manipulator, can also cause unknown defects in further generations. And if we look into the next class of therapy options, these are monoclonal antibodies, natalizumab, alemtuzumab, daclizumab, ocrelizumab, they target different receptors on the T and B cells, thereby dampening the immune system and inducing protection in multiple sclerosis. All of them are quite good in reducing again the annual relapse rates. They are actually taken as a next step or option by clinicians if the first generation DMTs do not work in an MS patient. So here again, owing to the fact that they dampen the immune system to a significant level, they come with a lot of infection risks. And for example, raclizumab has been called back from the market in 2018 owing to the fact that it caused severe brain inflammations, and PML infections are also quite common. Um, so ocrelizumab, for example, is proclaimed to work as a primary progressive multiple sclerosis treatment option, but of course there haven't been any clinical studies which prove the fact, and which now brings us to another option, which are off-label therapy options. For example, bone marrow transplantation, where a global immune cell reconstitution happens, or other chemotherapeutics like cyclophosphamide, azathioprine, methotrexate. All of them, they again reduce annual relapse rates to a significant level, and are good options for multiple sclerosis. And steroids, for example, act in an anti-inflammatory manner, thereby reducing the relapse rates, and are useful during pregnancy, but their efficacy is a little bit less compared to the other options. And here, when we talk about off-label options, there is also a fact. For example, in the case of cyclophosphamide, there have been no approved clinical trials which are proving its efficacy. Um, the trials, if some of them have been conducted, are again inconclusive. So all these are certain a lot of uncertainties, and there is a lot of scope to improve therapy options for multiple sclerosis. So now we move to the next step. Let us look at the pathophysiology of multiple sclerosis. Which areas can we further look to develop better therapy options? So as we see the brain of a multiple sclerosis patient, the myelin sheet around axons is destroyed, as you see in the videos, and this is the cause of their disease activity. And in a healthy brain, normally oligodendrocytes form the myelin sheet around the axons, which is required for the nerve impulse conduction in a quick and normal way. But when T cells are badly programmed by unknown factors, they cause release of cytokines which recruit other circulating immune cells like B cells, macrophages, and change their behavior. The badly programmed T cells also, when they cross the blood-brain barrier, enter the brain and release a lot of cytokines inside the CNS, thereby recruiting other resident immune cells like microglia and the entered B cells and macrophages. The microglia, the B cells on turn re release antibodies, thereby they cause damage of the axons, myelin sheet, and also the oligodendrocytes. All this damage further causes the collapse of the system completely, and the damage can also be visualized as lesions on an MRI. So this is the pathophysiology of multiple sclerosis. So here, which areas can we look upon to develop better therapy options? So as we know from literature that T cells, B cells cross the blood-brain barrier by various adhesion molecules like VCAMs, ICAMs, and B cells, for example, also use chemokine receptors like CXCR5, and there are certain treatment options in multiple sclerosis which target these proteins. This is a good therapy option, but of course there is a fact that we are blocking the immune cells from not entering into the CNS where they are required in certain cases like infections in the brain, for example PML or other infections. So there is again a compromise that we are making, that we are helping the patient, but we are keeping the patient again in the risk of developing some kind of cerebral infections. So what if we make or render the T and B cells tolerant to myelin? If we can utilize this strategy, we do not put the patient in the risk of developing any infections, because we leave his immune system intact, but at the same time we are modifying or acting only on the pathologic T and B cells, thereby preventing the damage to myelin. So here tolerance inducing therapeutics are under investigation, and three of the strategies I will be describing here. The first is peptide based, the second is DNA based, and the third is cell based tolerance inducing vaccination strategies. And under each strategy I will give a few examples. In the first one, peptide based method, peptides are direct from myelin origin, are directly injected into patients either transdermal or intradermal, and they cause an interleukin 10 secreting T cells, thereby causing the protective effect in multiple sclerosis. Significant reduction of annual relapse rates and a better MRI activity has been observed, but in cases of inflammatory state, this approach was not promising or did not yield any positive results. In the next approach, soluble synthetic peptides mimicking natural epitopes have been designed. They are called epitopes, and one of the examples is ATXMS1467, which is a cocktail of peptides from different myelin origins, and this, when injected into SPMS patients, SPMS meaning secondary progressive multiple sclerosis, these peptides have been tolerable and safe up to a dose of 800 microgram, and in phase 2A clinical trials in remitting multiple sclerosis patients, around 80% of them have shown less T1 gadolinium enhancing lesions. So this was quite a promising approach, and this could be followed up for scale up studies. And in the next approach, to further stabilize the system, liposomes carrying peptides of myelin origin have been used, and here as an example, mannose-related liposomes carrying peptides of myelin origin, which target the receptors of dendritic cells, thereby making them easily accessible to the dendritic cells themselves. So here CD206 targeting approach has been used, and these mannose-related liposomes have been easily taken up by dendritic cells, thereby causing an immune tolerance to the peptides in the liposomes. Here phase 1 clinical trials where patients from RRMS or SPMS patients of multiple sclerosis where they fail to respond positively to existing disease modifying therapies have shown a good profile of their chemokine system, thereby showing less inflammatory state when treated with these kind of mannose-related liposomes. So this again proves that tolerance inducing therapeutics are a better approach compared to the existing disease modifying therapies, and there is a great need to test this system in a large population, thereby evaluating its efficacy and safety too. And in the next example, this approach is more to extract peptides from T cell receptors of T cells which are pathogenic in multiple sclerosis. And as an example, Neurovax has been developed to a safe. It induced IL-1 secreting T cells, reduced myelin basic protein specific T cells, thereby these are the pathologic T cells, and in clinical trials they have shown promising results. And phase 2B trials are in process, and this approach is specifically developed for SPMS patients. And here again this approach could be very promising if it is scaled up to a larger level in multiple sclerosis. The next approach is based on DNA vaccines. These are vaccines which are developed, they code the complete um, uh, myelin protein of human origin, and as an example, BHT 3009, which is used in RRMS and SPMS patients, was shown to be safe and tolerable, and induced less gadolinium positive lesions, and interferon gamma producing myelin reactive T cells are also less in number. Autoantibodies are less in the cerebrospinal fluid. So it was quite promising in the initial levels of clinical trials, but in the phase 2, around close to 290 RRMS patients have again shown positive results, but the final stages did not show any benefit in the outcome of the disease. So this vaccine, although it was very promising in the initial stages of the clinical trials, requires further evaluation and probably a design much better in its um, clinical parameters should be tested with this approach. And in the next cell-based tolerance inducing vaccinations, the first one which we will talk about is T-cell vaccinations. And actually companies like Novartis, which is quite good in cell and gene therapies, T-cell vaccinations are actually in use in other disease areas, but not in multiple sclerosis according to my knowledge, of course. And here in this system, actually vaccines are developed by extracting T cells from patients and irradiating them and reintroducing them into the patient again, thereby causing a cytotoxic CD8 positive T cell response, and making them tolerable. But of course, this system, there are a few limitations in that the source and target are the same, and annual relapse rates are although reduced to a significant level in multiple sclerosis patients, and clinically the MRI was stabilized. Loss of efficacy and the source and target being the same are a few drawbacks of this approach. So to further fine-tune the system, T-cell clones targeting different myelin peptides have been targeted and developed, and this vaccine development strategy is a bit more broader in its approach, and Teclina is an example where phase 2b subcutaneous injections using such vaccinations in around 180 SPMS patients has been tried, but in this trial clinical endpoints were not met and the trial was discontinued. So to further stabilize the interaction between the peptides and the T cells, peptides of myelin origin and the T cells, chemical coupling has been used, and here there were promising results. In RRMS patients, close to 75% of T cell proliferations has been reduced, whereas in controls it was only a 30% reduction. There is a stability in the MRI activity of the patient. So this was quite good, and but the challenge is that this approach has been tested in a very limited number of patients, as I have shown in the presentation here, and there is a great need to test this system in a large population, and I believe this is a good approach. And in the next approach, so this is one of the most promising and most modern in my opinion too. Here dendritic cells are used as tools to develop vaccinations. The fact that dendritic cells are themselves antigen presenting cells, they have the ability to induce T cell energy and increase the regulatory T cell population, thereby they act as systems and good tools to develop vaccinations. And as an example, if we look, peptides of myelin origin have been pulsed to bone marrow derived tolerogenic dendritic cells, and when these are treated with vitamin D3, which is a biological, and they could induce protection in mouse model of multiple sclerosis. When applied in a preventive manner, the MS incidence was less, and when applied in post EAE application, the clinical improvements, the clinical parameters were improved. So there are 3 phase 1 clinical trials which are ongoing to check the safety and tolerability of this approach, and I believe there will be great results from these studies. So I have now finished this topic, and I'll be now entering into a different but very short topic, which I call a special note, because it is also very much related to my PhD topics. Here, as an example, I'll take a paper from a research group from the United States. They have described microparticles around 500 nanometer to be. They have designed them carrying peptides of myelin origin, and they were used as treatment options for multiple sclerosis in mouse models. But the group has described a prerequisite of Marco, which is a scavenger receptor on marginal zone macrophages, and regulatory T cells, and functionally active regulatory T cells, and T cell energy as prerequisites for the system to work. This group has also started a clinical trial after the publication in around 200 SPMS patients, and they are evaluating the safety, tolerability, and efficacy of this approach to be used as a vaccine in human beings. And I believe they will come up with great results in the coming years, and I would be happy to see how this proves to be useful in humans. And in the next 2 articles, the authors have described the role of calcium in myelin damage, how the different entry-exit mechanisms of calcium and the different cations contribute to the myelin sheath destruction and the oligodendrocyte destruction. This topic, I have taken this topic because some of my publications during my PhD time, and the main topic of my research was to understand cation, especially calcium, magnesium, and zinc entry-exit mechanisms in thrombocytes and immune cells, thereby their importance in thrombosis and stroke. This was my topic, and the reason why I am talking about this topic in multiple sclerosis is any projects or ideas which could be implemented or under plan to be developed for the treatment options of multiple sclerosis dealing with such topics, I could be of great use because of my background and my knowledge and the publications which I'll be now discussing. During my PhD, I was fortunate and to publish a few first author papers. The first one is about TRPM7 kinase, which is an alpha kinase linked to a channel conducting calcium, magnesium, and zinc. So here we could describe the role of this kinase in the different signaling mechanisms of thrombocytes and how it is responsible for calcium signaling, thereby its importance in thrombosis and stroke using mouse models. In the next article, we could describe the role of magnesium transporter MAGT1 and how this magnesium transporter and the magnesium conducted across it is responsible for the different calcium signaling pathways across various routes in B cells, and thereby its importance in the B cell receptor signaling. And in another first author publication, we could describe the role of zinc as a cofactor useful for coagulation, and how human and mouse platelets with different storage pool disorders could be evaluated for their defects or any other functionally inactive systems, and why zinc is responsible in such systems. So by these publications, I wanted to demonstrate that scientifically I am quite capable to conceptualize projects and to conduct experimental research and evaluate the results and thereby put them together as publications. So which is one of the primary requisites for a medical or clinical profession, and I wanted to demonstrate that I am quite well trained, and I have also demonstrated uh, my success during my PhD period. So taking this point again, I have great research experience in thrombosis and stroke areas, and additionally after my PhD I was for almost a year trained as a clinical research or drug safety associate in my further education and training program in a CRO from Berlin, and I now have currently this job too. I'm working as a sales representative for multiple sclerosis products in Novartis, employed by a third party organization. And from my origin I come from India, so I speak a lot of Indian languages, Tamil, Hindi and Telugu being my mother tongue. And because of my time for almost 10 years in Germany, I'm very fluent in German language, emails and also communication, and basically all of my education has been in English right from my childhood. And I also wanted to demonstrate my speaking ability through this presentation. So I'm very good in all of the 5 languages, and any global position requiring multilingual, multicultural skills, I would be of great use in such positions. And I can guarantee you that once given an opportunity, because of my cultural diversity and also my ability in science and research, and also now with sales and how to deal with customers and to understand their needs, so all these are my capabilities, and I would like to really utilize them and be very resourceful to the company that gives me an opportunity. And why specifically am I interested in Novartis? It would be a little bit of exaggeration, but I'm truthfully stating this point. It is Vas who is a great inspirational person, and I have observed and I've learned a lot from his videos which are present online. I'm unfortunate that I couldn't meet him till now, but if I get an opportunity I'd be very happy to meet him. And how he is transforming Novartis based on its 3 pillars: inspired, curious and unbossed. So these are the primary factors which interest me to work for Novartis. And additionally, the mutual interests in that the company Novartis is being driven by digital data science and artificial intelligence and platforms like cell and gene therapies. These are also mutual interests of me and the company, because I believe that only through digital and data science and artificial intelligence, problems of the healthcare system can be addressed in a very quick and rapid manner comparing to the conventional or traditional methods. And in addition, there are huge operations in India from Novartis, with close to 5,000 associates working from India. And owing to the fact that I come from India and I have a great cultural sensitivity and knowledge how things work in India, and with almost 10 years in Germany, as I stated before, so I'm a kind of hybrid national having both backgrounds, and I'd be very helpful to companies like Novartis, where functional roles requiring communications with India and European countries. So in such positions I would be very resourceful, I can guarantee you this. And in addition, the openness in Novartis through platforms like One Digital, where projects could be conceptualized, and the freedom that we get to express our thoughts and to be innovative, and other digital platforms like Yammer, where people share their knowledge, and this is a very good platform, and I have been greatly benefited the past few months through these 2 channels. And the vision of becoming a carbon neutral company by 2025, which is a very tough task, but still Novartis has promised to be such a company. So these kind of points interest me a lot to work for Novartis, and the story could go on. I can talk for hours together why I would like to work for Novartis. So if I get an opportunity to meet you in person, I would be very happy to talk a little bit more on this. And for now I took a lot of time, so I'd be very very happy to uh, hear from you. If you give me an opportunity in any of the um global positions where I could be resourceful in both communication with different countries, and to be very frank, and a global position is really meant to be a global position only when people can communicate in a global manner, who have experienced a different cultural level in their career and life. And I can guarantee you, with my profile and my background and my experiences that I've had till now, I can be a very good resource in a global position. Thank you very much, and I'd be very very happy to hear from you.